Assalamu alaikum and welcome. This tutorial is the continuation of our previous lecture. In our previous lecture, we have studied about photosynthesis and we gained knowledge how plants use carbon dioxide and water to form glucose. And in this tutorial, we will follow how these sugars are again converted into carbon dioxide and how energy is produced by our respirations. Respiration is the process by which we use the product of photosynthesis. We have already known about this, and it is the basic mechanism by which we extract energy from food molecules for usable forms. There are two types of respiration that are generally considered. One is external respiration. External respirations mean exchange of carbon dioxide and water, carbon dioxide and oxygens between atmosphere and human body. Human body take oxygen from atmosphere and give off carbon dioxide to outside of the environment. So it is called external respiration. And internal respiration is a cellular respiration in which the C-chain molecule like sugars, glucose, etc. are converted into their respective products, and energy is gained by breaking down of the C-chain compounds. There are various type of cellular respirations. We have aerobic respirations which occur in the presence of oxygens, and it is and complete breakdown of glucose only occur in the presence of oxygens. And other type is anaerobic respirations. It occurs in the absence of oxygens and it is incomplete breakdown, and we gain a little amount of energy with anaerobic respirations. Then we came across the process. The glycolysis means splitting step of glucose. Glucose is broken down into pyruvic acids. It is common to aerobic and anaerobic respirations. The process is that glucose is converted into the two molecules of the pyruvic acid plus some kind of energy, and NAD is converted into NADH2 and it is reduced, and so glucose is oxidized to the pyruvic acids. So we can say it an oxidation process. There are two type of aerobic respirations in which oxygen is used, and anaerobic respiration in which oxygen is not used. It is also called fermentations. Fermentations we can further be categorized into the alcoholic fermentation, in which alcohol and carbon dioxide are produced inside the body of the yeast, inside the body of bacteria, and the lactic acid fermentation, in which lactate or lactic acid is formed inside the body of living organisms like in the skeletal muscles when they perform a rigorous activity and they perform in the presence of little quantity of oxygen. Then our muscles do anaerobic exercise and lactic acid get accumulated in our muscles and they become taut and painful, and this is also the fermentations called lactic acid fermentation. We will get through this type of fermentations next. Here are the basics respirations types. We know pyruvate is formed by glycolysis process, which is common to both aerobic and anaerobic respirations, and what is dealt with. And this pyruvic acid, either in the presence of oxygens or in the absence of oxygens, is the real game. What is happening and where you decide whether it is aerobic and anaerobic respirations, and the product decide whether it is lactic acid fermentation or ethanolic fermentations. And with oxygens, like it goes to acetyl coenzyme A and enter into Krebs cycles, then it goes into the electron transport chain and provide a lot of energy to us, like 36 ATP, or net gain in eukaryotes 38 ATPs, and net gain in prokaryotes, and total gross product is equal to 40 ATPs. And then Without oxygen, there are two types: lactic acid fermentation and lactate is formed, and the ethanol alcoholic fermentations, in which first pyruvate is decarboxylated and converted into the acetaldehyde, and then it is converted other type reduced into the ethanol. These are the two basic types of aerobic or anaerobic respirations. And then we came across here is the aerobic respiration, and the pyruvate is converted into the ethanol is the alcoholic fermentations, and here is the lactic acid fermentation, and with the pyruvic acid is converted into lactate, and NADH2 two molecules are oxidized in this process, and these are the chemical reactions that are important. Then we came across the aerobic respiration. Aerobic respirations will discuss in. First of all, glycolysis is occur in cytosols, then all other process of the aerobic respirations occur in mitochondria, and mitochondria is basically involved in production of ATP from the glycolysis. So mitochondria is also called the powerhouse of cell to us. Then we came up the role of mitochondria. We will gain knowledge about this in further details. This is organelles that distributed throughout the cytoplasm of animal and plant cell, and it has crucially and internal membrane that are helpful in production of the ATPs by oxidative phosphorylation, by Krebs cycles, and many more reactions. And there are battery of enzyme and coenzyme that help in generating ATPs. So it is called a powerhouse of cell. Then adenosine triphosphate is the energy currency that is produced by mitochondria. Actually, organic food or inorganic foods is converted into the chemical form of energy, that is the ATP, that supplied energy to various type of muscular contractions, like nerve conductions, and various type of metabolic activities. And it is a high yield energy molecules. Phosphate, it contains three phosphate molecules, and two phosphate bonds are high energy, and when these phosphates are broken down, the maximum amount of energy is extracted. Like one phosphate is broken down and it is converted into adenosine diphosphate, then 7.3 kilocalories of energy is laid down. And it is then we came across anaerobic respiration. Only about 2% of energy present in the chemical bond of glucose is converted into the APTP. So as it is a less energy source and a poor energy source, and aerobic respiration's maximum amount of energy is gained. Then we came across biological oxidations. We need energy and biological birds, and various type of oxidation being addition of oxygen and other compounds. Removal of hydrogen is also oxidation process, and cellular respiration is essentially an oxidation process. It is the most important skills and often asked in APM and GC questions in MDCAT. So you have to study. There are various further steps of the cellular respirations, that are glycolysis, pyruvic acid oxidation, then it is entered into the mitochondrial membranes, and there is a Krebs cycle in mitochondria, and respiratory chain is also in a mitochondrial internal membranes. And so, and by various steps, energy is produced in the form of ATP. We will know how this is converted. First step is glycolysis, which occur in the cytosol. We'll study about glycolysis. Here are the detail of glycolysis and the complete pathway of glycolysis, how glucose is converted into the two molecule of the pyruvic acids. This is the complete 10-step process that here are the forms that we have. First of all, glucose, which is converted into glucose 6-phosphates. At carbon number 6 of the glucose phosphate is attached, it means glucose 6-phosphate, and 1 ATP is used. Here 1 ATP is used, and it is energy. Like we can call it energy. It is the process in which energy is used, and it is called preparatory phase of the glycolysis. Here is the preparatory phase of the glycolysis. Glucose is converted into glucose 6 phosphate. A phosphate is added at carbon number 6 of, then isomerizes, convert into the fructose 6 phosphate. It is the isomer of glucose 6 phosphates, then further phosphate group is added, fructose 1 6 bisphosphate. Phosphate group is also added at carbon number 1 of the fructose, then it is converted into the fructose 1 6 bisphosphate, then 1 6 bisphosphate is converted and degraded into the two molecule of three carbon compounds, dihydroxyacetone phosphate DAP, and glyceraldehyde 3 phosphates, and both are the phosphorylated, and both contain one phosphate group that was originally taken by the fructose molecules, then there is further phosphorylations of bis for dihydroxystone phosphate and glyceraldehyde phosphates. They are converted into the bisphosphoglycerate, and bisphosphoglycerate are glyceraldehyde 3 phosphate or bisphosphoglycerate. And there is also oxidation step in which NADH converted into NADH, and bisphosphoglycerate is the oxidized product. Then on a further step, ATP is produced, and phosphate attached to the 1,3-bisphosphoglycerate is used, and 1 ATP is produced, and 3 phosphoglycerate molecules are produced. You know that 1 ATP molecule is produced by this bisphosphoglycerate, and second is produced by this, and 2 ATPs are produced in this way. Then this is isomerizes, converted into 2 phosphoglycerate. Like at first phosphate is attached was attached, the ATP phosphate that is used in formation of ATP is phosphate attached at carbon number 1 position of the phosphate, and the phosphate that is attached to carbon number 3 is still present. Then it is converted onto the carbon number 2, and these are converted into the 2 phosphoglycerate molecules. And then there is the removal of water molecule, and phosphoenol pyruvate molecule are converted. Then the phosphate group that is still present in it is also given to the ADP, and ATP is produced, and two molecules of the pyruvates are formed. So the two molecules are pyruvates, each containing 3 carbon atoms, is subjected to the oxidations, and they are converted into the acetyl coenzyme A by specific type of enzymes and specific type of reactions. There are also the reduction of NADH and the oxidation of the pyruvate. The cycle begins when two carbon units from the acetyl coenzyme A react with 4 carbon molecule oxaloacetate to form produce citrate. There is also the decarboxylations of the pyruvate, and so it is left only with 2 carbon atoms, and this 2 carbon atom acetyl coenzyme A react with our precursor substance, which is oxaloacetate. Here is our 4 carbon compound oxaloacetate, which is reacted with acetyl coenzyme A and converted into the citrate, and one hydrogen H2 is added, and coenzyme is also recycled and are reused for formation of and for further pyruvate oxidation. Then citrate is converted into its isomeric form, isocitrate, containing 6 carbon atoms. Here is it is further oxidated and decarboxylated also. Like here, NADH are produced and carbon dioxide are released, and it is converted into alpha ketoglutarate of 5 carbon compound. Then same step is repeated with it, and it is converted into the 4 carbon compound 16 and the meanwhile series of the reactions, the thing that happen, GDP plus phosphate is converted into GTP, and when GTP converted into GTP again, then ADP is converted into ATP, and one ATP molecule is produced. Then further the fumarate succinate is converted into the fumarate. It is further oxidized, and now oxidized the oxidizing agent which is reduced is FAD, not NADH, and it is converted into the FADH2. And then fumarate is converted into malate by addition of water molecules. Then the next last step is malate is converted into our precursor xaloacetate by further oxidation. These type of molecules that are produced, NADH, NADH, FADH2, NADH, these are the donor of the hydrogen atoms, and when these hydrogen atoms is moved down the respiratory chains, this is used in formations of a particular type of ATP molecules. One NADH is used in production of nearly 3 ATP molecule, and FADH is used for production of two ATP molecules. There are total 3 NADPs and DH are producing in the Krebs cycles, and 2 FADH are producing in the Krebs cycles for 1 pyruvate molecules. And if we consider the 2 pyruvate molecule of a glucose that are obtained by it, we can double this calculations. Here was our Krebs cycles completed, and there was 2 ATPs were used in glycolysis, and 4 ATPs were produced. The net gain of ATP and a glycolysis is 2 ATP, and total ATP generated are 4 ATPs. You have to consider it. Then it is the last step, the respiratory chain. The NADH that are produced are the hydrogen donors, and they rotate and they slow down their hydrogen atoms on the respiratory chain to produce the variety of the energy in the form of ATP. And there are the respiratory chain components are coenzyme Q, series of cytochromes B, C, A, and 3. A and 3 are called A complex molecular oxygens, where it is at the end of the molecule and a final receiver of the electron formation and help in formation of the water and ATP. Then NADH is converted into NADH, and hydrogens and electrons are produced. And this step that gives 2 electrons gives so much energy. It is high in energy, and it come to low energy, and its energy is so much that it is forming the production of ATP molecule hairs. Then it is further oxidized by coenzyme Q, and FADH2 is also oxidized by coenzyme Q. And FADH, if we consider FADH is here, it is from next to 2 ATP, and we consider NADH here, it is from 3 ATP. So FADH is from 2 ATPs, and it from 3 ATPs. And if we consider cycle for this, it will start from coenzyme Q, and which is considered cycle for NADH start above this. Then coenzyme Q is further oxidized by B, and it produces hydrogen atom. Then B is oxidized by C, and this produce ATP. We have to remember that at what particular type and what particular positions of reactions ATP are being produced, then it is oxidized by A3. Then it reaches the molecular oxygens, and H2 is produced, and further ATP is produced. And for NADH, for the whole step, 3 ATP molecules are produced, and we consider FADH, then there is 2 molecules of ATP are produced. And we calculate the ATPs for pyruvate molecules. Then for Krebs cycles, then we have 3 NADH, and that will produce about 9 ATPs, and FADH that will produce by 2 ATP, and 11 ATP are being produced by pyruvate. And Krebs cycle further produce a 1 ATP, and so one Krebs cycle produced 12 ATP in total. and we have to repeat that Krebs cycle twice for a glucose molecule because it produces two pyruvate molecules, so it produces about 24 ATPs, and there was two ATPs produced by our two ATPs produced by our glycolysis, and 24 ATP plus 2 ATPs, these are the 26 ATPs, and there were NADH, one NADH produced by oxidation of the pyruvate, and it will produce about 3 ATP, and for other pyruvate it will produce about 3 ATP. So the total amount of ATP is almost about 36 ATPs that are being produced. Some here, some ATPs produced here, some ATPs produced by these NDH. These are the 32 ATPs that are produced by the NDH, by the NDH of Krebs cycle, ATPs of Krebs cycles of both pyruvates, and oxidation of both pyruvates, and directly from glycolysis. And there are these two NADH molecules that will produce about 6 ATP, and total 38 ATP are produced in prokaryotic cells, and 2 ATPs are used in the entering of pyruvate acetyl coenzyme A into the mitochondrial membrane. So it cost about eukaryotes 2 ATPs. That total net gain of the ATPs in the eukaryote is 36, because here the NADH is converted into ATP, and so it produces one less ATP, and for the both pyruvate it produces two less ATPs. So for one pyruvate there are 18 ATP produced by the eukaryotes, and 19 ATPs produced by prokaryotes. And if we consider the total gain of ATP, then we have to consider these four type of ATPs that are being used in above. Then we total amount of ATPs produced in a Krebs in a glycolysis is 40 ATPs, but we have to consider it 38 because 2 ATPs are used in glycolysis both in both prokaryotic and eukaryotic, because in both prokaryotic and eukaryotics the glycolysis occur. Then respiratory chain, here is the respiratory chain, then oxidative phosphorylations, in which production of the ATP occur in the presence of oxygens. And here is the complete diagram. One NADPH we will form 3 ATP molecules in oxidative phosphorylations. Here also the chemiosmosis coupling of the membrane occurs, but here the membrane is of mitochondrion membrane, and coupling is in the inner mitochondrial membranes, that is in the form of crystal. Like these are the differences between the coupling. Like coupling in the photosynthesis was in from stroma to lumen of the thalocid membranes, like because photosynthesis occur in plants only and not in animals, so the plastids and chloroplasts occur in only plants. Pumping mechanism across the inner membrane of mitochondria folded in crystal between matrix of mitochondrion and mitochondrion membrane matrix. Here is the matrix of mitochondria, and here are the its two membranes, inner membrane and outer membrane. Here coupling across the inner membrane of mitochondrion. Here is the complete summary, summary of the glycolysis or complete respiration process, and here are the important information which products inhibit which type of reactions? Like glucose is converted into fructose phosphofructokinase, fructose 1,6 bisphosphate, and converted into pyruvate at the end of respiration. Then it is oxidized, then it enters into Krebs cycle, then it into electron transport chain, and ATP are produced by all these processes, and these inhibits the fructose 6 phosphate is converted into phosphofructokinase, by which fructose is converted into fructose 1,6 bisphosphate, and this is enzyme is inhibited by ATP, and ADP NADH that is produced inhibit the pyruvate carboxylase. More the NADH that is produced, less will be activity of the pyruvate decarboxylase. NADH inhibit this, but these are the types of chemical reactions activate the phosphofructokinase, like citrate, like Krebs cycle. Here is the complete summary of the respirations. Hope so you have understand it.